Hello, good evening. So this video, it's going to be the notes from the professor over the synthesis of fatty acids. Last time we covered beta oxidation. These are going to be the parts that the professor highlighted for the synthesis of fatty acids. Okay, so to perform the synthesis, we would need acetyl-CoA as well as this molecule, malonyl-CoA. We would also need the NADPH, which is from the pentose phosphate pathway. Synthesis of fatty acids is going to take place in the cytosol or the chloroplast of plants. We are going to be primarily using the acyl carrier protein as well as this fatty acid synthase, but more on that later. There's usually 1 ATP needed every 2 carbons added, so there is an energy cost there. So the activating group will be 2 different enzyme sulfur hydrogen areas. It's not going to be the SH of CoAsh. So unlike beta oxidation where the activating group was the SH of CoAsh, this time it's going to be 2 SHs on 2 different enzymes. Okay, so first step will be to activate, not activate per se like phosphorylate anything, but we will need to make that malonyl-CoA. Malonyl-CoA is a 3-carbon molecule, and how you make it for this process will be taking acetyl-CoA and bicarbonate, that's HCO3, and then you're going to again need ATP for this. You need acetyl-CoA carboxylase. Carboxylase will be the enzyme, acetyl-CoA carboxylase, and then you're going to form the malonyl-CoA. This occurs in the cytoplasm. You'll need this biotin prosthetic group bound to a lysine on the carboxylase for it to work. The HCO3 will be carried by biotin, and you're going to have this lysine group perform this kind of swing in the enzyme when it's working on the acetyl-CoA and then forming the malonyl-CoA. Okay, so that's basically like the priming step. Now we actually talk about the main enzyme, fatty acid synthase. This will use a 4-step sequence in the cytosol. Each cycle will add 2 carbons to this new fatty acid, and you're going to start with acetyl-CoA and our malonyl-CoA. FAS2, or fatty acid synthase 2, is found in plants, vertebrae, and bacteria. It's dissociated, and each step is performed with a separate enzyme. However, in FAS1, fatty acid synthase 1, that's in mammals, there are 7 active sites in separate domains in a single multifunctional chain. There are 2 chains, and they're independent, however they act as homodimers. And you get a single product in FAS1. So we have the MAT transferase domain. This is going to need the correct CoA molecule. Acetyl-CoA's acetyl group will go to the ACP, the acyl carrier protein, and then it's going to go to this other region called KS. So why do we couple condensation with decarboxylation? And that's going to just be the 3-carbon molecule plus the 2-carbon molecule. And you get a 4-carbon molecule with CO2. If we go back up here, I think I wrote down which is which somewhere. Oh well, I don't see it right now. Okay, never mind. Oh, malonyl-CoA is a 3-carbon. Okay, so malonyl is the 3-carbon. So this must be acetyl-CoA. I'm guessing. I don't know. Highly exergonic. This release of the CO2 is highly exergonic. Okay, so step 1, these are the actual steps of fatty acid synthesis. So condensation is the first step. The malonyl-CoA will go to the acyl carrier protein, the ACP. The acetyl-CoA will go there too, but then it's going to move to the KS. Okay, so once that happens on our fatty acid synthase 1, then we're going to get acetyl-acyl ACP plus CO2. The CO2 again is being released. The acyl carrier protein will shuttle the acyl group to another site, which we saw. And carbons 1 and 2 are going to be acetyl-CoA. We're going to be working on the carbonyl group of the acetyl-CoA, and that's tied to the malonyl-CoA, but the malonyl-CoA, it's bound elsewhere. Okay, so second, we're going to use reduction. We're going to take the C double bonded to O group of the acetyl-CoA, and we're going to turn that into a CH2. This is going to be a beta-ketoacyl ACP reductase, the enzyme that will be used in this. And we also need NADPH in step 2. Step 3, we're going to use a dehydration reaction to get an alkene. We're going to get an alkene with this at the ACOA, at the acetyl-CoA. So the enzyme for step 3, the dehydration, will be B-hydroxyacyl ACP dehydratase. Number 4 is another reduction. We're going to be using enoyl ACP reductase, and this alkene will become an alkane, also known as butyryl ACP. We also need NADPH at this step. So step 2 and step 4, you require NADPH. So basically, condensation, reduction, dehydration, reduction. All right, so now we transfer that butyryl group to the KS area as number 5. Number 6, step 6, we're going to reattach a new malonyl-CoA. We're going to recharge a new malonyl-CoA to the ACP. And then step 7, once we've gotten enough carbons, remember we're adding 2 carbons per go of this. And so that means we will be, at one point, we're going to have to separate the chain from the ACP. So we're going to be doing that. Step 7, and I forgot the enzyme that does that. Let me look it up quickly. Sorry about that. I should have wrote it down. I think I planned to, but it just like slipped my mind, or may have shipped off somewhere else in the page. I'll find it later on. Sometimes that happens, but not a lot. It's not on this drawing app for sure. Sorry about that. This is gonna cost me some time. Okay, step 7 will be thioesterase. So this is a thioesterase. I should write that. So thioesterase is step 7. You separate the chain from the ACP region. Okay, so after that, for palmitate, a 16-carbon fatty acid, you would get this reaction here. 7 acetyl-CoA, 1 CO2, and ATP for 7 malonyl-CoA at the beginning. Then the overall reaction will be 7 cycles of condensation, reduction, dehydration, and reduction again. That will be acetyl-CoA plus 7 malonyl-CoA plus 14 NADH. Oh, NADPH. Sorry about that. 14 NADPH. And that's going to make palmitate. And therefore, the overall reaction of palmitate will be this right here. 8 acetyl-CoA plus 7 ATP plus 14 NADPH will equal our palmitate. Okay, so biosynthesis of fatty acids requires acetyl-CoA, which we've been talking about. You need that for formation of the malonyl-CoA. Let me go back up here. Sorry about that. Yeah, you need that for malonyl-CoA formation. And you also need it down here, the condensation reaction. Okay, so we know that ATP is needed for the malonyl-CoA synthesis. Remember that acetyl-CoA carboxylase, you need ATP for that to make malonyl-CoA. So that's why we have ATP. It creates the group transfer potential. Okay, so we also need the reducing power of NADPH. Why do I keep writing? There we go. NADPH is needed in steps 2 and 4, which are the reduction steps to reduce the beta keto group and the double bond. And I think, are those in order? Yeah. First, you're going to reduce the beta keto group, and then you're going to do the double bond. All right, so fatty fatty acid synthesis is a cytosolic process. So, however, in plants, again, this is going to be in the chloroplast. This allows for the segregation of cofactors for beta oxidation and synthesis. And fatty acid synthesis occurs where NADPH levels are high. Okay, so how do we make this NADPH? So we talked about the pentose phosphate pathway before, where we're going to be taking glucose 6, and then it's going to go through that process, make a ribose, and then NADPH. However, we also have this malic enzyme plus malate, and that will result in pyruvate plus CO2 at the same time as creating some NADPH. Alright, the acetate is shuttled out as citrate. It's shuttled out of the mitochondria as citrate. So acetyl-CoA is formed in the mitochondria from pyruvate oxidation or the amino acid skeletons. And so there's like just this big like cycle. I'm going to just talk about it. So acetyl-CoA plus oxaloacetate in the mitochondria. We've seen this step before. This is in citric acid cycle. You're going to use a citrate synthase. That makes citrate. Citrate could go in to the cytosol. And then you have a lyase, and that's going to make oxaloacetate. Oxaloacetate will then be worked on by this malate dehydrogenase, and that's going to make malate, and the malate could go to the mitochondria. Malate, once it's in the mitochondria, we're going to have this malic enzyme, and then that's going to create some NADPH as well as pyruvate. So this is the key right here, NADPH, malic enzyme or pentose phosphate pathway. Okay, so fatty acid elongation systems, these are used to lengthen palmitate to form long saturated fatty acids. For example, this one right here, stearate, is an 18-carbon fatty acid, and that's made from palmitate. That's just an example of one. Desaturation of fatty acids needs oxidase, so we're going to be using a desaturase to do that process. Okay, so palmitate and stearate can be desaturated. However, humans cannot desaturate beyond delta 9, and that's just the position of the carbon bond. Eicosanoids are formed from 20 to 22 carbon polyunsaturated fatty acids. One of these would be arachidonate or arachidonic acid, and these hormones include prostaglandins, leukotrienes, and thromboxanes. The prostaglandin is actually highlighted, and you have this cyclooxygenase, or COX, and that's going to make the prostaglandins that regulate gastric mucin activity or secretion. So regulates the mucus, the gastric system, its secretion. The COX2, however, this will be dealing with the mediation of pain, inflammation, and fever. Therefore, aspirin and NSAIDs, these are going to block this COX enzyme via the serine acetylation. You're going to add an acetyl group to serine on these COXs, and that's going to stop them. Therefore, the serine, it could be any amino acid that has an OH group, so tyrosine or threonine would also work in that situation. So if we use aspirin, there's going to be no pain. However, we can't make any mucus. Right, so how do we regulate oxidation and synthesis? It's tightly regulated. One of the main parts is malonyl-CoA. Remember this priming step where we took acetyl-CoA, bicarbonate, and ATP, and we had the acetyl-CoA carboxylase to form malonyl-CoA. Well, the first intermediate, this malonyl-CoA of cytosolic fatty acid synthesis, it blocks the entry of the fatty acids into the mitochondria. So in this situation, we have a kinase and phosphorylation. If glucagon is present of the acetyl-CoA carboxylase, that will turn the enzyme off. High AMP, therefore, inactivate the acetyl-CoA carboxylase by changing it into these inactive filaments. However, if we have a phosphatase that removes the phosphate group of the acetyl-CoA carboxylase, and if insulin is present, that will turn this enzyme on, and we're going to be making malonyl-CoA. If malonyl-CoA is present, that will prevent the carnitine shuttle, so then we can't go into beta oxidation in the mitochondria. High citrate will then also, let me see, high citrate will also block the activity. Oh wait, no. High citrate will form, sorry about that, it will form malonyl-CoA. It will jump that reaction, and then therefore the synthesis of fatty acids will go forward. However, this is blocked by glucagon, high AMP, looks like. Okay, so because acetyl-CoA carboxylase is the rate limiting step, that means high citrate will then kick it forward. You're going to make the malonyl-CoA, and then you're going to have the synthesis of the fatty acids. Sorry if that wasn't clear. Okay, let's see. We are going to then, yeah, that'll be the next video. I was about to go to that, but that's amino acid oxidation. That'll be the next video. All right, thanks so much. Take care, and do something nice for someone.